आज हम डिस्कस करेंगे बैक्टीरियोलॉजी का एक टॉपिक, ग्राम पॉजिटिव बैक्टीरिया बैसिलस देखेंगे आज। इंट्रोडक्शन उसका देखेंगे। जीन्स बैसिलस, जो जीन्स है वो बैसिलस इंक्लूड्स, ये ग्राम पॉजिटिव बैसिलस में आता है जो प्रोड्यूस करता है स्पोर्स। ये जीन्स जो है ये साइक्रोफिलिक, मीजोफिलिक, थर्मोफिलिक स्पीशीज़ में आता है विथ मैक्सिमम टेम्परेचर जो है वेजिटेटिव ग्राफ्स के लिए इसको 25 डिग्री सेल्सियस टू अबोव 75 डिग्री सेल्सियस। मिनिमम कितना टेम्परेचर होना चाहिए? 5 डिग्री सेल्सियस टू 45 डिग्री सेल्सियस। ठीक, जो साल्ट टॉलरेंस है इसकी वो लेस देन हो सकती है 2% टू 25% NaCl। ठीक, their heat resistant spores, some which are killed only at 120 degrees Celsius, may be found in soil, water and air. ठीक, commonest contaminants in bacteriological culture media. अब देखेंगे Bacillus anthracis. The causative agent of anthrax is a major pathogen. Anthrax is primarily a disease of cattle and sheep and may affect other animals also. Humans get infected when they come into contact with spores from any of these sources. दूसरा है Bacillus stress can cause foodborne gastroenteritis. Some species may also be responsible for opportunistic infections. अब हम Bacillus anthracis देखेंगे डिटेल में। इज एरोबिक, ये एरोबिक है, ग्राम पॉजिटिव है, ये नॉन मोटाइल, नॉन एसिड फास्ट, स्पोरुलेटिंग बैसिलस है विच मेजर्स 1 to 1.5 into 3 to 5 माइक्रोमीटर साइज इसका होता है। अरेंज सिंगली और इन शॉर्ट चेन्स। द एंटायर, यहाँ दैन नहीं है, यहाँ द है। द एंटायर chain may be surrounded by capsule. Capsule is polypeptide in nature. Capsule production depends upon a 60 megadalton plasmid. In clinical material and culture, it appears in chains and has appearance of bamboo stick. अब हम स्पोर्स के बारे में थोड़ा सा देखेंगे। Sporulation occurs under unfavorable conditions. Oxygen is required for the formation of spores but not for their germination. ठीक, spores are highly resistant to physical and chemical agents. Spores are formed in culture or in the soil but never in the animal body during life. अब कल्चर कैरेक्टरिस्टिक्स देखेंगे। Bacillus anthracis is an aerobe and facultative anaerobe with a temperature range for growth being 12 to 45 डिग्री सेल्सियस। ऑप्टिमम टेम्परेचर क्या होता है? 35 to 37 डिग्री सेल्सियस। ऑन न्यूट्रेंट agar media, colonies are round 2 to 3 mm in diameter, raised, opaque and grayish white. Blood agar medium, the colonies are non-hemolytic. Through occasional strains produce a narrow zone of hemolytic. Gelatin state culture, gelatin is liquefied mostly at the top due to aerobic environment and gives a characteristic inverted free dry appearance. Biochemical reactions, glucose, maltose and sucrose are fermented with production of acid only, gas नहीं, acid बस। Catalyst यहाँ बनता है nitrites and are reduced to nitrites. अब हम pathogenicity देखेंगे। The virulence factors have been identified in pathogenesis. The virulence factor of anthrax bacilli depends on two factors: extracellular toxin, capsular polypeptide. The toxins consist of three components, namely the protective antigen factor, edema factor, lethal factor. Pathogenesis, anthrax is a zoonotic infection, animals are infected by ingestion of spores present in the soil. The disease is a fatal septicemia in animals but may sometimes be localized, resembling the cutaneous disease in humans. Infected animals shed large number of bacilli from the mouth, nose, and rectum. These sporulate in the soil and remains as the source of infection. Human anthrax is contacted from animals. The disease may be cutaneous anthrax, common site of entry of the infection, or the face, neck, hands, arms, and neck. Okay? This lesion is called a malignant pustule. This disease is also known as Hyde-Forster's disease. The disease used to be common in dock workers carrying loads of hides and skins on their bare backs. Pulmonary anthrax. This is called wool shotter's disease because it is used to be common among those who worked in wool factories where they would inhale spores from infected wool. The infection is characterized by hemorrhagic bronchopneumonia, often followed by septicemia. Intestinal anthrax. This occurs mainly in common communities that consume undercooked or uncooked meat. Severe enteritis with bloody diarrhea occurs, ending fatally if left untreated. Anthrax meningitis or meningoencephalitis. This is a fatal infection which occurs in the CNS via the bloodstream. अब देखेंगे हम लेबोरेटरी डायग्नोसिस. पहले देखेंगे स्पेसिमेन, स्वैब, फ्लूइड और पस फ्रॉम pustules, sputum and blood from pulmonary or septicemia anthrax are generally collected. Fecal specimens or peritoneal fluid may be collected from intestinal anthrax. अब माइक्रोस्कोपी देखेंगे. gram stained smear from specimen often show chain of large gram positive bacilli. Capsule appears as clear hollow around the bacterium by Indian ink staining. Direct fluorescent antibody test DFA for capsule specific staining and for cell wall antigen confirm the identification. McFadden's reaction: when blood films are stained with the polychrome methylene blue for 10 to 12 seconds and examined under microscope. Culture specimen is inoculated on nutrient agar medium and incubated at 37 degrees Celsius. Medusa head colonies appears on the medium. Smears made from these colonies show typical gram-positive spore-bearing bacilli. Gelatin tape culture show inverted fire tree appearance. ठीक. Animal inoculation: while mouse or guinea pigs are injected with exudate or culture, animal dies within 36 to 48 hours. Smear made from hard blood and sputum shows bacilli. Serology: Ascolis thermoprecipitin test. The tissue is grounded up in saline and boiled for 5 minutes and filtered. When this extract is layered over the anti-anthrax serum in narrow tube, ring of precipitate appears at the junction of two liquid within 5 minutes in a positive case. ठीक. पॉलीमरेज चेन रिएक्शन भी हम यूज़ करते हैं इसमें डिटेक्शन के लिए.